Hi, this is Maddie Gallerson, and I will be presenting on CRISPR-Cas9. This will cover an introduction of background information, the theoretical basis and methodology, which covers the basic principles and how it works, advantages and disadvantages of CRISPR-Cas9 gene editing, and then applications focusing on a specific paper. So we're going to cover some biographical information on first part. So Jennifer Doudna and Emmanuelle Charpentier are the two women credited with harnessing the CRISPR-Cas9 system for gene editing. Jennifer Doudna was born in Washington, D.C., but grew up in Hawaii. She ended up at Pomona College for her undergraduate degree and then Harvard for her graduate degree in biochemistry, where she worked with RNA and DNA. In 2002, she moved to Berkeley and continued to study the structures and function of RNA. In 2005, she heard about a rare bacterium with clustered, regularly interspaced short palindromic repeats, CRISPR, and decided to try and figure out how it seemed to be able to mount a defense against previous attacks. The other woman in this duo is Emmanuelle Charpentier, who was born and raised in Paris and got her undergraduate degree in biochemistry from Marie Curie University and then received a PhD in microbiology. She later went to the Pasteur Institute for her graduate research and investigated genetic and molecular mechanisms of antibiotic resistance. Later, her focus turned to how pathogenic bacteria interact with their hosts and environment and developed some tools to facilitate genetics and bacterial pathogens and studied proteins involved in adhesion and signal transduction. She later went to New York before coming back to Europe and establishing her own research group at the Max F. Perutz Laboratories. While there, she became interested in regulatory mechanisms in bacteria that involved small RNA molecules and developed the CRISPR-Cas9 project. In 2011, she published a paper that identified the role of tracer RNA in CRISPR-mediated defense. And then in that same year, she met with Jennifer Doudna, and they started to work together on harnessing the CRISPR-Cas9 system for gene editing. So the development of this method is a little bit interesting since it already exists in naturally occurring bacteria. So rather than created, it was adapted and harnessed for gene editing. So when a bacterium is infected by a virus, it captures a part of the viral DNA and incorporates it into the bacterial DNA in an area known as a CRISPR array. This allows the bacteria to recognize and attack the virus if it were to infect the bacteria again using a Cas9 enzyme, which would disable the viral DNA. The researchers realized that this system could be adapted for splicing and integrating specific DNA sequences into like a cell's genome. And so by using guide sequences made from short RNA sequences, they're able to target specific DNA portions, so genes, using the Cas9 enzyme. This enzyme is capable of cutting the DNA at a targeted location, just like it is able to cut viral DNA in a bacteria. And after the DNA is cut, the researchers can then add, delete, or make changes to DNA sequences. Some previous methods of gene editing include zinc finger nucleases, transcription activator, like effector nucleases and meganucleases. Zinc fingers are segments of a protein that are able to recognize and bind to specific DNA segments. These natural zinc fingers have led to ZFNs being used as DNA cutting enzymes. There's a therapeutic company that has developed ZFNs from laboratory tools that could actually disable disease-causing genes or correct errors in existing genes. A transcription activator like effector nucleases are made of DNA-binding proteins that recognize a DNA sequence fused to a nucleus effector domain to achieve cleavage of the DNA. Tails are secreted by bacteria and contain a conserved series of residues with two divergent amino acids. These divergent amino acids are responsible for determining the DNA binding site specificity by choosing combinations of repeat segments. So while the applications of ZFNs and TALENs overlap, the TAKENs have an ease of design because of their recognition code. Then there are meganucleases, which are nucleases with long DNA binding recognition sites that can be up to 40 nucleotides long. The length of these nucleases makes it unlikely that natural sites would be present by chance in a complex genome. The challenge with the meganucleases is that there is difficulty in designing new ones for target sequences of interest. Some success has been achieved, however, they are unlikely to be used much in the human genome given simplicity of other methods. The next part of the presentation is the theoretical basis and methodology. Looking at this slide, we see a picture of the gene editing done by the CRISPR-Cas9, and we see the Cas9 and guiding RNA, which have bound to the matching DNA sequence and have done a double-stranded blunt cut. Underneath, we see the two different options for end joining slash repair, which is a non-homologous end joining and a homologous direct repair. These can either lead to the deletion of a gene or the insertion of a gene, depending on what the researcher wants. In the next slide, we see biological and biochemical principles. And so this kind of breaks down what we're seeing. So the guide RNA is a programmed piece of RNA that is meant to direct the Cas9, which is an endonuclease, meaning that it cuts in the interior of a DNA sequence. And so this directs the Cas9 to the target gene. The Cas9 endonuclease then binds to the guide RNA and then creates an effector complex, which is now able to recognize the protospacer adjacent motif, which is the PAM. When it finds the PAM, is able to bind to it and then checks that the DNA and the guide RNA are complementary. If they are complementary, it is able to make a blunt double-stranded cut. If they are not complementary, it will continue to look for that complementary section at the PAM. It is very specific, so unless there is an issue with the guide RNA, it is going to find that PAM and cut in the targeted DNA sequence. And then there is the break and repair. Once the cut has been made, and it is a blunt cut, meaning there are no sticky ends, it is just the nucleases lined up with each other, then the DNA has to be repaired. It can't leave the break there. This is where the researcher would be able to add or take away a gene or DNA sequence from the genome, and this could be used for seeing what a new gene would do within a cell, or for maybe knockout therapies where you're seeing what would happen if a gene was not in the cell. However, this also can lead to certain issues, which we will talk about in the next section of advantages and disadvantages. As hinted at, the next thing is the advantages and disadvantages of the CRISPR-Cas9 gene editing. So some advantages are that it is easy to disrupt a targeted gene. So for those knockout therapies that we're talking about, you can easily get in there and disrupt the gene and keep it from being translated and transcribed. You're also able to insert new sequences at precise spots within the DNA with the use of that RNA guide. And so you're able to be very precise with that. And it also reduces the time and expense of developing models, since you can just go ahead and do it. And then also, it's relatively cheap and easy to create these guide RNAs, and also does not require very much training in how to actually use the CRISPR-Cas9 complex. Some disadvantages are it is difficult to deliver material to mature cells in large numbers. CRISPR is not something that is going to be shared from cell to cell unless it's in a germline cell or a stem cell. And so you would have to get it to all the adult cells, which is very hard to do. And then that second disadvantage is that even when the cells do take in the CRISPR-Cas9, they may not actually have any genome editing activity. So even if they take it up, there is no guarantee that the genome editing will actually take place. And then off-target edits can occur, and they have severe consequences. Say if you were to turn off a regulatory gene or a housekeeping gene, you could cause potential harm to the cell or the organism. And then with any kind of gene editing, there are always going to be ethical issues and concerns. And not only does this affect the usage of the gene editing software, but it also affects the research, considering that people are afraid of the potential uses. So while there are many applications of the CRISPR-Cas9 gene editing software, we'll be talking about the specific application as used in this paper entitled CRISPR-Cas9 mediated PD-1 disruption enhances anti-tumor efficacy of human chimeric antigen receptor T cells. Specifically talking about, within the paper, the generation of PD-1 deficient human CAR T cells with Cas9 RNPs. And so you'll see in the picture, this is the protocol that they used for CRISPR editing and lentiviral transduction. So what they did was they used the CRISPR-Cas9 and the guide RNA effector complex to disrupt the PD-1 protein on the T cell exterior. by doing this, they can interfere with the protein, which is part of programmed cell death or apoptosis. And by interfering with the apoptotic pathway, they can boost the autoimmunity of the T cells. And T cells are incredibly important to the immunity and immune system of cancer patients. And that is what this is specifically focusing on, considering that it is dealing with anti-tumor treatment. And they did find that using the CRISPR protocol, that they were able to create these PD-1 deficient cells. So this paper shows that the CRISPR editing and lentiviral transduction being used to create these PD-1 deficient human CAR T cells could potentially increase autoimmunity and would have anti-tumor efficacy with these chimeric antigen receptor T cells. And this could be helpful for treating both benign and malignant tumors, since it increases T cell activity as well as the activation of T cells, since it knocks out those PD-1 receptors and interferes with the apoptotic pathway in these chimeric antigen T cells.